Miss Venezuela, what does it take to be a Miss Yanna-verse? Miss Venezuela wishes to respond to the question, but her English is limited. Can you tell what is lacking in this frame? That's right, a translator who is fluent in English and Spanish. As the question was asked, a translator should have been there, decoding and translating the question to Miss Venezuela to help her perform the question and answer portion flawlessly. This reminds me of one of the most important terms that Francis Crick has coined, the central dogma of molecular biology. Have you ever wondered how DNA makes RNA and how RNA makes protein? This fundamental concept is called the central dogma of molecular biology. It explains the transfer of genetic information in cells. But how does it work? Since the DNA contains instructions required for making proteins, it needs to be copied before cell division, so that each new cell receives a full set of instructions to operate properly. This process is what we call replication, but our focus will be on the two steps of protein synthesis, transcription and translation. Transcription begins as the enzyme RNA polymerase binds and opens up the DNA helix. Complementary nucleotides are then added to the template DNA strand. In the same way that DNA is used as a template during replication, it is also used when the mRNA transcript is made, which detaches from the DNA strand. Afterward, a cap of the methyl group is added to the 5' end, while a poly-A tail is added to the 3' end. These modifications provide stability and protect the mRNA from degradation. Subsequently, the primary mRNA undergoes splicing wherein introns or intervening segments are removed, while exons or protein-coding regions are reconnected, forming a mature mRNA, which is set to release in the cytoplasm. This is where the next step begins, translation. Translation involves decoding an mRNA and using its information to build a polypeptide or a chain of amino acids. It begins as the ribosomes assemble and attach to the mRNA. Next, the mRNA is read and the anticodon of the tRNA binds to the codon of the mRNA. The amino acid of the tRNA is then added to the growing protein chain. Once a stop codon appears in the ribosome, the chain is separated from the tRNA and expelled from the ribosome. In addition, there are 64 possible codons and 20 amino acids. Out of these codons, 3 are stop codons, namely UAG, UAA, and UGA, while 1 is a start codon, namely AUG.
Muchas gracias.
Thank you so much. Ladies and gentlemen, our Miss Yanna-verse 2022. This has been Yanna Balita, your junior biologist, leaving you with a lesson on the central dogma of molecular biology. Genetic information flows from DNA to RNA to protein.